A warm welcome to my channel. Today's topic is glycolysis and overview. In this video, I will give a quick overview of what glycolysis is, and the details of each step will be shared in the later videos. Glycolysis is also called the EM pathway or Embden-Meyerhof pathway after its discoverers who elucidated this in muscle cells. Glycolysis has been derived from two Greek words glykees and lysis, where the former means sweet or sugar and the latter means splitting. So basically in glycolysis, one molecule of a 6-carbon compound named glucose is splitted into two molecules of a 3-carbon compound called pyruvate using 10 enzymatic steps. The free energy which is released during this process is entrapped in the form of ATP and NADH. Glycolysis is the first metabolic pathway to be elucidated. Its importance: glycolysis is almost universal central pathway of glucose metabolism and is the sole source of metabolic energy in various mammalian tissues and cells, including erythrocytes or RBCs, renal medulla, that is the medullary region of kidney, brain, and sperm. Moreover, anaerobic microbes are entirely dependent on glycolysis for the formation of metabolic energy. There are plants that store starch like potato tubers and some aquatic plants like watercress which are dependent on glycolysis for their energy. Now, as I said, glycolysis is a 10-step process. It is divided into two phases of glycolysis. The first 5 steps are called the preparatory phase, whereas the last 5 steps contribute to the payoff phase. Now this is the preparatory phase, wherein phosphorylation of glucose and its conversion to glyceraldehyde 3-phosphate occurs. It is a 5-step process and involves the use of these 5 enzymes: hexokinase, phosphohexose isomerase, phosphofructokinase, aldolase, and triose phosphate isomerase. Here, glucose is irreversibly converted to glucose 6-phosphate with the utilization of one molecule of ATP. Glucose 6-phosphate is then converted to fructose 6-phosphate, which is again phosphorylated at first carbon to form fructose 1,6-bisphosphate with the utilization of one molecule of ATP. Fructose 1,6-bisphosphate with the action of aldolase is splitted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These two molecules are interconvertible via the enzyme triose phosphate isomerase. So in the preparatory phase, two molecules of ATP are utilized and glucose is splitted to form glyceraldehyde 3-phosphate to dihydroxyacetone phosphate. In the payoff phase, oxidative conversion of glyceraldehyde 3-phosphate to pyruvate and the coupled formation of ATP and NADH occurs. As I said, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are interconvertible via the enzyme triose phosphate isomerase. But the formation of glyceraldehyde 3-phosphate is preferred. So we get two molecules of glyceraldehyde 3-phosphate from one molecule of glucose. This glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate via the enzyme glyceraldehyde 3-phosphate dehydrogenase with the production of two molecules of NADH. 1,3-bisphosphoglycerate is then converted to 3-phosphoglycerate with the formation of two molecules of ATP. 3-phosphoglycerate with the help of phosphoglycerate mutase is converted to 2-phosphoglycerate, which upon the release of two molecules of H2O result in the formation of phosphoenol pyruvate. Phosphoenol pyruvate is irreversibly converted to the final product of glycolysis, that is pyruvate, with gain of two molecules of ATP. So in the payoff phase, we get 2 plus 2, 4 molecules of ATP, and in the preparatory phase, we lose 2 molecules of ATP. So the net gain of ATP molecules is 4 minus 2, that is 2. All these steps will be discussed in detail in the later videos. Thank you. Please like, share, and subscribe my channel for more videos.